Well, we'll get on with introducing Duncan Murdock, who's going to be our speaker this evening. Duncan is based at the Natural History Museum in Oxford. Has been there for quite some time, and he's going to give us a talk this evening on evolution's first predators, I believe, the origin, evolution and ecology of the first vertebrate predators, conodonts. Yeah. So I'll leave it to you then.
Okay, thank you very much. So thanks everyone for coming out on this slightly damp evening. I appreciate you making it out. So yeah, thanks for the introduction and the invitation to speak. I am a local lad. I grew up around here. My mum and dad have come this out the front. So it's a great pleasure to speak to this group because I owe an awful lot to this area and the amazing geology that's here in the Black Country for kicking me off on my career. And I also owe a lot to Graham, who's at the back as well, for setting me on the path towards museums. So yes, thanks very much to Graham too. I also want to thank a bunch of collaborators and co-conspirators who have contributed to this research. One of them, Paul Smith, is also here, who's lurking in the back. So if I get any of this wrong, it'll be Paul who's heckling me to correct the mistakes because he's contributed to some of this research as well. So yeah, a number of students and advisors, collaborators, people that have provided specimens for this research. But first of all, a little bit about, briefly about me and what I do now. So I have the great privilege of working in this fantastic building. This is the Oxford University Museum of Natural History. So opened in 1860 as this cathedral to science, the home of science in Oxford University. So I've been in Oxford for about 7.5 years, first as a research fellow and then later and currently as a collections manager. So in Oxford we are very proud of our fantastic collections. Some notable examples include the Hope Entomology collection, which is perhaps the best collection of British insects in the UK. We have the only soft tissues of the dodo surviving anywhere in the world up there, and the first dinosaur ever to be named, Megalosaurus. We've been celebrating the 200th anniversary of Megalosaurus all the way through this year. We also have absolutely beautiful neo-Gothic architecture, and art and architecture on the inside of the building is worth seeing even before you've looked at anything that's in any of the cases. It's still breathtaking every time I walk in the building. So if you haven't made the trip down to Oxford to see us, I highly recommend it. It's free, open almost every day of the year except for Christmas. So please do, if you're ever in that part of the world, come down and see us. So my role in the Earth collections, I look after our rocks and minerals and parts of our fossil collections. So we have about 40,000 minerals, about 150,000 rocks, give or take. The rock collection's hard to count because if you drop one, you end up doubling the size of that particular collection, and almost nobody will notice it, so that's not too much of a problem. Normally I wouldn't say that in front of the director, but he's retired now, so I can say what I like. And we have about 500,000 fossils, strengths in the Pleistocene, the Ice Age of Oxfordshire, Jurassic Oxfordshire, but also lots of fantastic Silurian fossils, including many from are local to here. I'm very privileged to be involved in our displays as well. So here, this is me installing a meteorite in one of our brand new displays. So as well as looking after collections, I get to do brilliant public engagement. So here is me holding one of our beautiful Dudley bugs. So as a I took this picture quite early on in my role as looking after these fossils, because the 8-year-old me would never have believed that it would be my job to look after the fossils, some of the fossils that made me want to be a paleontologist. So much so that I even dressed as a trilobite once. This is a photo that doesn't get aired very often. So there's me dressed up as a trilobite. My enthusiasm for paleontology, as I said, has a long pedigree. So here's 8-year-old me getting excited about dinosaurs. Hopefully my fashion sense has slightly improved since then, but you can blame Mum and Dad for giving me the bum bag and the sunglasses that I'm sporting there. So that's just a little background into my interest in the subject. So the rest of the talk, I'm going to talk about my favourite group of fossils, the conodonts. So this, I have to confess, this is not my image, but this is a scanning electron micrograph of the head of a pin with 4 conodont elements on the top, just to give you an idea of how small these things are. So they're these tiny tooth-like elements that come in this amazing array of shapes, but they're almost entirely tiny. They're super abundant. If you get rocks of the right age, marine rocks of the right age, you're almost guaranteed to find loads of conodonts. They're absolutely super abundant, but relatively poorly known group outside of certain paleontological circles. They're very useful fossils. So they have this remarkable property of changing colour depending on the temperature that the rocks they're buried in has reached, the so-called conodont alteration index. And they're also fantastic biostratigraphic tools. You can tell where you are, and this is an example of one of many biostratigraphic papers. I picked this one because it features our very own Graham Wharton as a co-author. And just to show you that there are conodonts right here as well in the lower Paleozoic rocks in the Black Country as well. So until the middle part of the mid to late 1980s, what kind of animal these conodonts belong to was something of a mystery. They've been suggested to be almost every kind of organism that you can think of, everything from plants to mollusks. But a discovery was made in in the Granton shrimp beds. So these are carboniferous rocks from the foreshore just outside Edinburgh. They have these beautiful shrimp fossils with soft tissues preserved. And just shy of a dozen conodont animals, the bodies of the conodonts were discovered and described. So they have this head, which is where you find those tooth-like elements, this long eel-like body, a big pair of eyes at the front, a set of muscles running down the body, and a tail that extends beyond the end of the gut. So all of these are characteristics that tell you that this animal is a vertebrate, effectively a fish, the group of animals that we belong to. Subsequently, another site was found to preserve the soft parts of conodonts from the Ordovician, this time of South Africa. And I was very privileged to work on some fossils from this location as part of my master's research that I couldn't fit into this talk. But again, we see these are muscle tissue, muscle fibers preserved from the body of this animal. And the other thing that fossils like this and fossils from a few other places in the world preserve is all of these elements still preserved in situ as they were in life. So over here, these two sort of spheres are the eyes of this animal, and then you have this array of tooth-like elements that would have formed this sort of basket that it was using to capture its prey. So we're able to reconstruct a lot about what these animals were like from these vanishing rare fossils of the whole body. This is a recent reconstruction that we've been working with an artist to do some artwork for some of our displays of what we think that conodont animal looked like. So we've got these two eyes. You can just about see these little white tooth-like elements, gills running down the body, these muscle blocks, and this tail fin. So this is what we think these creatures look like. But very tiny, just a few. The biggest ones are a few tens of centimeters, but most of them are typically just a few centimeters in length. But nevertheless, they were the scourge of the plankton. They were visual predators that were swimming around in the oceans. So where do they sit in relationship to other organisms? So this is a family tree of the vertebrates. So these branches in the tree represent different groups of fishes and how they're related to each other. Just so you know, we are here. We are in this group called the lobe-finned fishes, along with all of the other tetrapods. We're just fish that decided to walk around, but essentially just fish. The conodonts are here. And the other thing that's on this diagram is time. So present day is over where I'm standing, and 600 million years ago over on the other side of the room. So the conodonts originate around about 508 million years ago, towards the end of the period known as the Cambrian. They were one of the most abundant and diverse group of vertebrates all the way through the Paleozoic, all the way to the end of the Triassic, when they met their sad demise and went extinct. You'll notice that most of the branches in this tree are split into two to show that one group is more closely related to another group than anything else. But if you look down at the base of the tree, we've got three branches. And that's because it's not clear whether conodonts are more closely related to fish with jaws, like us and sharks and most of the other thing that you think of when you think of as fish, or jawless fishes, hagfish and lamprey. But they sit in that part of the vertebrate tree. Okay, so I want to talk to you today about two different pieces of research that I've been able to do on this group of animals, the conodonts. One as part of my doctoral research in the University of Bristol, and the other as part of my research as a research fellow in Oxford with Paul. So we're going to first of all talk to you about the first set of research. So here's another one of those trees. It's the other way up, so apologies for that. You'll have to twist your head round. So now time is going up the diagram. So the oldest things are at the bottom, 540 million years ago, and up to the Silurian. So the rocks from Wren's Nest are there in the Wenlock near the top of this diagram. And this is a tree just of a select group of conodonts. So most conodonts that we find are these so-called complex conodonts that go off in this up this arrow and evolve into a whole range of different forms through the Ordovician and Silurian. But the very earliest conodonts that we find in the latest Cambrian and in the early part of the Ordovician are these so-called coniform or cone-shaped elements that appear to have a much simpler morphology, and very little is known about them and their relationships to each other and their ecology and what they were doing. So this is a particular group of conodonts that I'm particularly interested in. So not only am I interested in an obscure group of fossils, I'm interested in the most obscure ones of that obscure group of fossils. So, but anyway, it's dinosaurs is far too crowded, so it's much better to be working on primitive conodonts. And in particular, there's this group right at the base of this tree called the paraconodonts that are a particular mystery. So for a long time, we've known about these three groups of cone-shaped teeth that appear in the Cambrian: the protoconodonts, paraconodonts, and euconodonts. Now, the protoconodonts were originally considered to be among the rest of the conodonts, but if you compare them to a group of marine worms called the chetonaths, or arrow worms, which I'm sure very few people have probably come across. They're a group of modern marine worms that swim around eating plankton. They have very similar shaped elements that they use to grab hold of their prey. So it's now believed that protoconodonts are nothing to do with vertebrates. They are chetonaths. So we can cross those off the list. But that leaves us with these two different groups, the paraconodonts and the euconodonts, and the question is, how do they relate to each other, if at all? So here are two elements of the small one is a paraconodont, the big one is a euconodont, and I picked these, so that's 100 microns down there, so that's a tenth of a millimeter, so you can see there's a very tiny. I picked these out of a sample with a paintbrush and a microscope. Then they come from exactly the same piece of rock. They both come from the very latest Cambrian of Nevada. So we know that these things were coexisting, but are they relatives of each other, which we wanted to find out. So it all boils down to how they grew and what's inside these elements. So this is a diagram of a euconodont sliced in half, so you can see the layers that built up as it grew. And there's three questions that we wanted to answer that hadn't been understood. The first is, what happens to the very oldest part of the element? The first part that grows, which is highlighted in yellow. The second is, how do the layers on the inside of the element relate to the layers on the outside? And the third is, how do the two different kinds of tissues that we see, so this dotted tissue, which we call the basal body, and this tissue up here that we call the crown, how do they relate to each other? So to try and answer this question, I was lucky enough to get to do a technique called SRXTM. It's synchrotron radiation X-ray tomographic microscopy, but you don't need to worry about that. Just think of it as a very, very, very big microscope. So the smaller things get, the bigger the microscope you need to look at them. This is, in fact, an entire building, donut-shaped building. This one is in Switzerland, near Zurich. So this is a photo from the website of the Institute. So here we have this group of learned continental scientists off to get their cafe latte to discuss particle physics or whatever they were interested in. So this is what I expected to find when I got to the Swiss light source where we were. Of course, I was a lowly doctoral student, so this is not what I saw. This is what I saw. So the machine runs 24 hours a day, and if you're the new boy, then you get the night shift. So quite often, this is what I saw on my way into the building. But once you're inside, we go into this little hutch, to this particular instrument that was called Tomcat. So Tomcat gets the award for the worst acronym ever. So Tomcat stands for Tomographic Microscopy and Coherent Radiology Experiments. Okay, I didn't come up with that. And then once inside there, so this is a model of the whole facility. Essentially, it's a donut-shaped tube that you fire electrons down and then bend them with big electromagnets. And as they fall around this tube, they release energy in the form of X-rays. And you channel those X-rays down a tube, which is called a beamline. And you put your object of interest in front of that and rotate it. And then just like taking an X-ray of a broken bone, you look at how the X-rays are absorbed by by the object in front of the X-ray and capture those images. And because you've rotated it through 180 degrees, you can put all of that information back together and build a 3D model. So it's also equivalent to a CAT scan. If any of you have ever had a CAT scan, you get wheeled into an instrument. So if you go to a medical CT scanner, the camera and the X-ray source spin around you, because people tend not to like being rotated through to 180 degrees. Conodonts don't mind, so we were able to keep the X-ray source still and the camera still and rotate the specimen, but otherwise it's exactly the same. So here is what it looks like on the inside. So this tube here is where the X-rays come down. Here is me inserting a specimen. So you can see that you've got a camera up here, and then a mirror here which reflects the light and captures the information that you mount in a little pin. And then you sit outside and shut a very big thick door, because you don't want to be irradiated, and gather the data on a computer like this. So by doing that with these early conodonts, we were able to literally look inside and reconstruct how these animals grew. And so this is one single paraconodont element. And what I've done here is colored in each of the growth layers, separated them out, and you can see how this element grew from the very earliest part in D through to the most mature part in H. And the thing I want you to take away is that all the way through the life of this element, the yellow part, the sort of goldy yellow part, which is the earliest part of the element, is still visible at the tip of the element. It's not overgrown by any of the subsequent layers. And so we were able to therefore answer what happens to the very earliest part of the element during the growth of these conodonts. The next thing we did was put a bigger element in with lots more layers. So again, I've colored in the first few layers, and you can still just about see that yellow tip is still visible even in F. The other thing we can do is have a look at these layers as they grow around the base of the element. So to save you from squinting, because I've colored in a single layer, and hopefully you can see that it wraps around the base of the element. So these layers start on the side, on the outer side, and then they wrap all the way around the base, and then they go up the other side. So again, we're able to describe the geometry of those growth layers precisely. I also looked at euconodonts that have this second tissue, so that which differentiates them, and was able to do the same thing, look at the layers in the basal part, and then wrap over the outer part. So hopefully you can see this green part of the basal body is entirely engulfed by this red crown tissue. Okay? But if you take off the crown, then what you're left with is exactly the same as the paraconodont that I just showed you. So euconodonts are paraconodonts with a tissue added onto them. So here we can see a side-by-side comparison of a paraconodont with just the blue tissue, and a euconodont with the blue and the red. And the inner part, the basal part of this euconodont is almost identical in terms of how it grows from a paraconodont. So this allowed us to build up a model of how these elements grew. So we've got a cartoon version of the model here, showing that, and then build up a step-by-step evolution from the very simplest paraconodont elements all the way up to the euconodont elements that we see. So we were able, by using this technique, to answer this question of how paraconodonts and euconodonts are related to each other, in that paraconodonts are just euconodonts that haven't evolved this second tissue yet. So we were able to solve this mystery. So we wanted to take, that's why we can then draw on this tree that it's around about here, somewhere in the upper Cambrian, where we get the evolution of this tissue and a transition from one group to another. So we wanted to take this a step further and ask why. Why did conodonts bother when they were already building pointy teeth from one set of tissue? Why grow a second tissue? So to do that, we wanted to look at, well, how would these different teeth perform when they're feeding? So we can't take a conodont and have it bite something because they are sadly extinct, but we can do the equivalent of that digitally with a computer simulation. So we took elements like this one, took a cross-sectional profile, which we were able to derive from this data that I described we got in Switzerland, and then demarcate the outer edge and the boundary between the basal body and the crown, and then convert that into a simple silhouette, and then fill those in with a series of simple shapes. In this case, they're triangles. So we're doing that because to work out the physics of a really complex shape is very, very hard. But the physics of a triangle is relatively simple. So if you do the maths for lots of triangles and add them all up together, then you end up you're able to understand what's happening to a complex body. It's called a technique called finite element analysis that we've borrowed from engineers. So this technique is why planes don't fall out of the sky and bridges don't fall down, because you're able to test in a computer how these complex structures react to different stimuli. So then in a computer we grab hold of the base of the element so that it can't move, and then push against it at the tip to simulate feeding. And then we give the two different tissues slightly different material properties, slightly different stiffnesses. So your teeth have dentine and enamel. The enamel is very, very hard. The dentine is softer and more elastic. So we used similar material properties, in fact those of a shark, to simulate how these would react. So then we did that for paraconodonts and euconodonts that were similar in shape. And you build up these patterns of how stress, so that's how the bending of these elements as they function is accommodated through the different element. And we can do that with other conodonts of different shapes and build up a different picture. Oops, sorry, let me go back. So what we were able to find is that in paraconodonts of similar shape, the stresses build up at these pinch points, and the brighter, redder colours are higher stress values. So they would be potential points of weakness during function. Whereas in the euconodonts, the stress is much more evenly distributed all around the outside of the element. So we're able to say, not only did euconodonts evolve this tissue, we can postulate why. It was to give them, make them function better as teeth. So they were better accommodating stress during function. So by throwing all these techniques at these conodonts, we're able to learn an awful lot about the evolution of this group that's nearly 500 million years old. So they might be obscure, but we can still learn an awful lot about them. Okay, so in the second half then, if I just take a drink. I'd like to talk you through a piece of work that I've done more recently on another group of another coniform conodont. So if we go back to that tree, so the very earliest conodonts are these paraconodonts with these cone-like teeth just made of a single tissue. Then we see the first euconodonts evolving a crown. And then there's this group here which is the other group of conodonts that make it through this interval and proliferate in the Ordovician and the Silurian that maintain this relatively comparatively simple cone-shaped teeth. And they were relatively understudied and relatively poorly known compared to their more diverse cousins on the other side of the tree. So I was very lucky to be able to examine this fossil. Apologies, the scale bar is missing, but this is just a couple of centimeters long. You'll see it with a scale bar shortly afterwards. So as I alluded to earlier, the soft tissues of conodonts are incredibly rare. There's about a dozen specimens from the Carboniferous of Scotland. There's the one that I told you about from the Soom Shale. And then there is this. This is from the Silurian of the US. So this is, and these are the soft parts down here, and here are the elements up here. So I was very privileged to be able to examine this specimen. So this was had previously been described in the late 1980s by Paul and colleagues by examining the elements that you can see on the surface of this specimen, and that allowed them to build up a picture of what this animal the elements in this animal might have looked like. This is a model that I reproduced in a paper that I wrote with some colleagues in 2013. So a series of these curved cone-like elements arranged in this apparatus. And this is essentially the only known specimen where we have the soft tissues along with the apparatus for this group of early conodonts. So everything else we know are these more derived complex things that we see later on. So we wanted to see if we can, with the range of tools available to us now that weren't available 30 years ago, if we can re-examine this fossil and try and find more about it. So here are some close-up photos of this specimen. So it's in this layer of rock that when you split it open, some of the elements go on one half and some go on the other. So that's what you're seeing here. You're seeing part and counterpart of the same specimen. And hopefully you can just about make out these little curved cone-like elements. Some of them are preserved as impressions in the sediment like these. Sometimes they're preserved intact. So here, these are microphotos. So again, the scale bar here is 1/5 of a millimeter, 200 microns. So you can see they're very tiny little elements. So we're able to take light photographs like this. But I was also able to generate a 3D model of the surface using a similar technique. We actually did this with a CT scanner rather than a synchrotron, but it's the same kind of technique, X-rays, penetrating the rock and building up a picture of what's inside. So this is exactly the same as what you just saw, but this is a 3D a rendering of that surface. So hopefully already you can already see more of the elements appearing because we're able to take out the color, exaggerate the shadows a little bit more, and see the surface of the rock. So as was already identified, there were 11 elements preserved on the surface. So we can then isolate those elements and color them in separately to try and look at how they're arranged with each other. And hopefully you can see that some of them are sort of diving into the surface of the rock. So where the rock is split open, the complete elements aren't visible because some of them are still buried in the subsurface. But because we've got this 3D data, we can peel away that surface and we can see parts of the elements that were otherwise buried. So we're now getting a more complete picture of the shape of these elements than you could see by just examining the surface. And we were also able to identify 4 elements that were entirely buried. So here they're labeled A, B, C, D. So they were completely unknown because they were not visible at all on the surface of the elements. So rather than 11, we've actually got 15 elements in this preserved in this specimen. The other thing we're able to do is artificially fill in those impressions. So some of the elements are not preserved at all. They're just voids in the rock. They're just little holes in the rock. So what I was able to do was digitally pour in some silicon rubber, if you like, fill in those gaps, and then isolate those molds. So the sort of ghostly parts of this image are the molds of those elements. So by combining what you can see at the surface, the molds, and what's subsurface, we can build the most complete picture of this apparatus that we've ever been able to see, and see the geometry of 15 of those elements. So there they all are. So we can then digitally break that apparatus apart in a way that you can't do with the real specimen without making the curators very, very cross. So we're able to see that we have pairs of elements, right and left sets of elements that I've coloured with the brighter and dimmer colours. So you can see element 1 and 2 form a pair, etc., down the apparatus. So we can then look at this complete apparatus and compare it with isolated elements that have previously been described. So here is a set of elements from this animal, which is known as Pandarodus. So we can find which elements we have preserved. So if we zoom in on this part of the elements that we saw, we can identify which elements are which. Now, you don't need to worry about the details of which are which, just that we're able to do that from the geometry of these elements, the shape of what's preserved. And we can do the same with the back half of the apparatus. And that allows us then, once we've identified which are which, to say, well, where are they? Are they where they were thought to be from the previous set of data that was described? And we can update this hypothesis of how these elements sit together. And we were able to do just that, which I'll show you in a moment. So we wanted to put this in the context of what we know about other conodonts. So at the top here is perhaps the best preserved apparatuses of conodonts from Bailey Falls, from the Carboniferous of the US. Researchers were able to build a very complete view of how these elements were arranged in space. And they built up this picture of how these elements were arranged. And it was thought that coniform conodonts were arranged a little bit differently. Arranged rather than being the elements perpendicular, they're arranged parallel to each other, facing each other across the midline. But how do you go from an apparatus with parallel elements to an apparatus with perpendicular elements? Well, there's two different options of how you can rotate those elements in space. And by characterizing the apparatus of Pandarodus with more detail than was able to be done before, we were able to answer that question and work out how you evolve from a parallel architecture to a perpendicular architecture by rotating the front part outwards rather than inwards. So it might seem like a subtle distinction, but it matters in terms of the function of these elements, why they were evolving in the way that they did. So as well as identifying the elements, as I said earlier, we have soft tissues of this animal, which is even more exciting because it's even more rare. So we wanted to use a variety of different techniques to characterize these soft tissues. So what you're seeing here on the to your left, go backwards, there we go. This is a scanning electron micrograph, so a close-up image using an SEM of just the elements. And what we're able to do with an SEM is also identify which chemical elements make up the conodont elements. Just realize it's going to get confusing if I keep saying elements. But so this sort of teal color is phosphorus. So Conodont elements are made up of calcium phosphate, just like our teeth and bones. So it's not surprising that we see phosphorus overlapping where we find these conodonts. But hopefully you can see that not all of that teal blue colour is coincident with the elements. Some of it is appearing in these white patches that aren't part of the conodont elements themselves. So we have phosphorus preserving outside of the elements. So we've interpreted this as the soft tissues that were supporting the elements themselves, the actual cartilages that were holding those elements in the jaw. So conodonts, unlike us, had no bones, so they have no jaw for those teeth to sit in. Like jawless fishes, they're sitting in soft tissues. Modern hagfish and lampreys have a range of cartilages that allow these elements to function, allow them to be predators, to go around and acquiring their prey. They become very specialized to particular kinds of feeding in the oceans, but they do this by having cartilages. And even the specimens that I showed you before from South Africa and Scotland, there's no cartilage preserved associated with the elements. So if we're right, and I'm very happy to be challenged on whether or not we are, but if we're right, we've got the first evidence of conodont cartilages associated, so we can make direct relationship with modern hagfishes and lampreys. So outside of the apparatus of elements itself, there is this slug of tissue that represents the body of this animal. So again, from the original descriptions, you're able to see quite a lot of information at the surface, but we know that there's information buried beneath the surface of the rock. So we were able to extract that using these X-ray techniques. So again, so we've got the elements up at the top. We've got in white at the top is that phosphorus map showing you where the cartilage is. And now we have this big area of turquoise that is also calcium phosphate. So this dark outline here is what was visible at the surface, and this grey part here is what we can see if you add together the subsurface as well. So you can see we have much more of the body preserved than was previously visible. And we also see much more detail than was visible before. So again, this is an X-ray slice. If you chop just below the surface, you can see I've chopped through some of the elements, and you can see their basal cones. The dark grey stuff is the host rock. Then this is calcium phosphate, and then these bright spots is pyrite, iron pyrite. So it's much denser to the X-rays. That's why they're showing up as these bright spots. So hopefully you can see that they're concentrated in and around the body tissues, particularly in the central part of this body. If I put the phosphate back on, you can see in the gaps between sort of arcs of tissue, we've got this pyrite. So that'll become important in a second. The other thing we can do is manipulate this in any orientation that we like, in ways that you can't do with the fossil. So we're now looking at it at a sort of oblique angle, and hopefully you can see these sort of banana-shaped structures running down the bodies. These are blocks of muscles, just like if you're running down our body, or if you fillet a fish, you'll see these muscle blocks that make up the body of this animal that's been preserved in calcium phosphate. So in one of my other, with a slightly different hat on, when I was a researcher at the University of Leicester, I did some decay experiments. So these aren't actually mine, but these were from one of my colleagues. I continued some of his work. So at the top are some of these muscle blocks of a primitive kind of animal, close relative of vertebrates, called the Amphioxus. At the top it's fresh, and at the bottom it's decaying. And hopefully you can see you've got these arrowhead-shaped structures that have started to spread apart and leave gaps in between them. Sorry, let me go back. Just like we see in the body of Pandarodus, we have these arrowhead-shaped structures with these gaps in between. So I think what we're seeing here is rotting muscle tissue. So we wanted to try and investigate that a bit more and have a look at the chemistry of those different tissues. So again, using a scanning electron microscope, we can ask it at different points, what is the abundance, the elemental abundance of different things that we can see. So don't worry about the graphs. I'm going to colour it in. So the grey is phosphorus, which I've already described. You have calcium phosphate. Red is iron. Green is sulfur. And where you have sulfur and iron, it's yellow. So all these bright yellow spots are iron and sulfur, and I'm sure I don't need to tell you that's pyrite. So those are those little spots of pyrite. Pink is where we've got iron and carbon, and blue is carbon, and so aquamarine is sulfur and carbon. So hopefully you can see you've got the phosphate, and then a region around it, which I've marked with an H, which is full of these bright yellow pyrite spots, and then a region which I've marked in M, where the iron is not reacted with sulfur, where you've got iron oxide. And this little box here is a close-up of the phosphate, and you can see these sort of string-like structures. So they should be, if you can remember back to the start of the talk, the fossils from South Africa had very similar looking string-like structures in the calcium phosphate. So more evidence that these are muscle tissues. So what we interpret this as is we have the muscle preserved as calcium phosphate, and then immediately around that, all the iron has reacted with sulfur because these muscles have been rotting for some time, and they've been releasing decay fluids into the sediment, which changes the chemistry of the sediment, and that gets as far as here. And then what we're seeing here is ordinary sediment where the iron is reacted to oxygen rather than sulfur. So we get iron oxides rather than sulfides. So if we have a look at the original fossil, we've got, this is the body, this is that decay halo, and then this is the rusty red iron oxides in the sediments. So comparing that with the animals that we see from Scotland, we had this narrow, long, eel-like body, okay, which is not what we see in Pandarodus. We see a much wider, flattened body shape. So we interpret this as an animal with a slightly different body shape, rather than being thin like an eel, which is a body that is adapted to swimming through the water. We've got this flattened, much more like a sort of flat fish, not quite as wide as a Dover sole, but this sort of more flatfish body shape that would probably be better adapted to swimming closer to the seafloor. But still as a visual predator, because it has this feeding element up at the front of the animal. So until we're able to describe in this level of detail the anatomy of the body of this animal, we didn't know how diverse the body shapes of conodonts were too. So we were also able to get a reconstruction of Pandarodus. This is one of very few public outings of this very new reconstruction. So we have this flattened body, these blocks of muscles that I showed you, and we have nothing from the tail preserved in the Pandarodus specimen that I showed you. Unfortunately, it's off the end of the slab. So we're interpreting it to have a similar tail fin to the other elements that we know. And then this fearsome-looking array of elements at the front. But please don't have nightmares, because it was only a couple of centimeters long. So only frightening if you're a Silurian zooplankton, that it was going for its dinner. So by applying a whole range of different techniques, we're able to piece together so much more about the anatomy, the ecology, and the evolution of this fantastic group of fossils. So I'll leave it there with this. This is a photo I took from the Smithsonian in Washington of a trio of conodonts swimming their way over a coral, looking to find some plankton to munch on. So I'm very happy to take any questions. Otherwise, thank you very much for your attention.
No more questions. I'd like to offer a vote of thanks to Duncan for coming along on this visual evening and giving us such a fascinating insight into the morphology and evolution of a fossil group that is probably not so well known to most people. So thank you very much. So I'd like to offer a vote of thanks
My pleasure.
to Duncan for coming along for this visual evening and giving us such a fascinating insight into the morphology and evolution of a fossil group that is probably not so well known to most people. So thank you very much.
No, thank you.